There are total 3 types of excretion that takes place depending on the availability of water and also the waste product which is eliminated. First is ammonotelism. What is ammonotelism? When the nitrogenous waste is excreted in the form of ammonia, the process is called as ammonotelism, and the animals which excrete ammonia are called as ammonotelic animals. They are basic in nature. Ammonia is basic in nature. Ammonia is soluble in water. It needs large quantity of water. So, the animals which have plenty of water around their surrounding, those excrete the nitrogenous waste in the form of ammonia. For example, 1 gram of ammonia requires nearly 300 to 5 ml of water for its excretion. Example, aquatic invertebrates, bony fishes, aquatic amphibians are ammonotelic. And without excretory system, example protozoa. The example protozoa also is called as ammonotelic animal. Excrete ammonia by skin, gills and kidneys. These animals which were given ammonotelic animals, they excrete ammonia from their general skin surface or with the help of gills or with the help of their kidneys. Second type is ureotelism. What is ureotelism? The nitrogenous waste which is excreted in the form of urea is called as ureotelism, and the animals which excrete the urea are called as ureotelic animals. Urea is less toxic form, it is less water soluble, so it requires less water for its solubilization. 1 gram of urea requires nearly 50 ml of water. Mammals, cartilaginous fishes, aquatic reptiles, these are the examples of ureotelic animals. Ammonia is converted into urea by urea cycle in liver, which requires 3 ATP molecules. Third type is uricotelism. When the nitrogenous waste is excreted in the form of uric acid, then the process is called as uricotelism, and the animals are called as uricotelic animals. Uric acid is the least toxic form. It is least soluble, so it requires minimum or no need of water for its elimination. Ammonia is converted into uric acid in the liver by the inosinic acid pathway. Examples of uricotelic animals are insects, reptiles, birds, land snails, etc. In animals which need conservation of water, usually they excrete uric acid. The next point is osmoregulation. What is osmoregulation? As we have already seen, that maintenance of solute and water balance in the body is called as osmoregulation. For example, now marine bird, example albatross, is usually found near the sea or on the sea throughout their life. Then what do they drink? Do they drink sea water? Yes, they drink the sea water throughout their life. Then how do they maintain their osmoregulation? They have the special gland called as salt gland which are present near their nostrils. These glands secrete salt by active transport, that is from lower concentration to the higher concentration with the investment of energy or with the investment of ATP. They secrete or they excrete the salt by active transport, and this way they maintain the osmotic balance of their body. Now there are 2 types of the animals. Either the animal or organism may be osmoconformer or the organism may be osmoregulator. What are osmoconformers? The animals which are iso-osmotic to the surrounding means they change their body temperature or they change their internal condition according to the environment, according to the surrounding, then they are called as osmoconformers. Example, marine organisms are usually osmoconformers. Second, what are osmoregulators? The animals or the organisms which can control their internal environment independent of the external environment are called as osmoregulators. Example, terrestrial organisms are usually osmoregulators. Whether osmoconformers or osmoregulators, animal need to tolerate a range of salt concentration. Then they either may be stenohaline or they may be uryl-haline. Who are stenohalines? The animals which can tolerate narrow range of salt concentrations are called as stenohalines, and uryl-halines are the organisms which can tolerate wide range of the salt concentration. Usually the true organs for excretion are present in the animal which shows bilateral symmetry. Next point is 2 major types of nephridia. There are 2 major types of nephridia. One is protonephridia and the second one is metanephridia. We just have to see what are the definitions and examples of the animals which contain those nephridia. Protonephridia are the network of dead end tubes which are usually called as flame cell. For example, in kingdom Animalia, we have seen that platyhelminthes have flame cells for excretion. Also, rotifers shows presence of protonephridia, few annelids have protonephridia, and amphioxus shows presence of protonephridia for excretion. Second is metanephridia. Metanephridia is the unbranched coiled tube that connects the body cavity, which is called as nephrostomes. Example are earthworm. Now the animals may excrete the waste products by their special glands. For example, Malpighian tubules are present in insects. Green glands are present in crustaceans. In mammals, for example, in humans, we have a special organ called as kidney which performs the function of excretion, and kidneys have the main basic and structural functional unit called as nephrons for the excretion. The next point is excretory system of humans. Excretory system includes two pairs, that is, The next point is excretory system of humans. Excretory system includes pair of kidneys, a pair of ureter, urinary bladder. Now we are going to see one by one. First is the kidney. Pair of bean-shaped kidneys are present which are situated from 12th thoracic to the 3rd lumbar vertebrae. Kidneys are usually called as retroperitoneal because they are situated behind the peritoneal membrane. The dimensions of kidneys are 10 into 10 into 13 centimeters. Weight of kidney is 125 gram in males and it is 135 gram in females. There are total 1 million of nephrons, that is the functional unit of the kidney, present in each kidney. Ureters. What are ureters? A pair of ureter arise from the renal pelvis, that is this point of kidney, from each side. So, a pair of ureter arises from renal pelvis of each kidney. There are the dimensions of ureters are 25 to 30 centimeter in length. It opens into the urinary bladder. The next point, that is the urinary bladder. These ureters open into the urinary bladder. The third point is urinary bladder. Urinary bladder are hollow muscular organs which are situated below the urethra. The triangular shape present at the base of the urinary bladder is called as trigone. Covered externally by the peritoneum. They are usually covered from outside by the peritoneum. There are total 3 layers of smooth muscles which are present in the urinary bladder. They are longitudinal muscles, circular muscles and longitudinal muscles respectively. The muscles which are present in the urinary bladder help the urinary bladder for stretching during the passing of the urine. Next is urethra. After the urinary bladder, there is a small tubular structure which is called as urethra, which open into the exterior of the body. Tubular structure open to the exterior. There are total 2 types of muscles which are present in the urethra. First is the internal muscles, second are the external muscles. Internal muscles are usually involuntary in function, that is their activity cannot be controlled by our own will. External muscles are voluntary in function, that is their activities can be controlled by our will. Now we are going to see the functions of kidney. What are the functions of kidney? First, we know that it is required for the elimination of nitrogenous waste and toxic material out of the body. Second, it maintains homeostasis. What is homeostasis? Maintenance of the constant body temperature, body conditions are called as homeostasis. This work is done by kidney. Third, it produces calcitriol and renin. Fourth, it secretes erythropoietin, which is useful for the production of new RBCs. Next, urinary bladder, it is also called as the storage organ of urine which temporarily stores the urine. It helps in micturition, that is passing off or the expelling out of the urine. Lastly, urethra, which is important for discharging of the urine out of the body. In males, this is also called as the urinogenital organ because in males it does the work of passing off of urine, semen and also sperms. The next point is the structure of kidney. There are total 3 layers of tissues which covers the kidney. The outer is called as renal fascia, the middle is called as adipose capsule and the inner is called as renal capsule. The outer layer, which is called as renal fascia, its work is the attachment of kidney to the abdominal wall. The middle layer, which is called as adipose capsule, its important work is it acts as a shock absorber. third inner layer, which is called as renal capsule, it helps from spreading of infection in the kidney. LS of kidneys shows two layers or two regions. The outer region is called as renal cortex, which is also reddish in color and it is a granule appearance structure. The inner is called as medulla region, which is pale red, that is slightly light red in color, and it is striated in appearance. If we see the structures which are present in outer and inner regions, then we see renal cortex contains Malpighian body. It also contains convoluted tubules which are called as PCT and DCT, that is proximal convoluted tubule and distal convoluted tubule, and blood vessels. These three structures are present in the renal cortex. The next region, which is called as medulla, contains two regions. One is the loop of Henle, that is U-shaped structure, and second is the collecting duct. The next point is structure of nephrons. Here the nephrons are 4 to 6 cm long, thin walled tube, and they are made up of single layer of epithelial cells. First part is the glomerulus. The glomerulus is a bunch of fine blood capillaries lying in the Bowman's capsule. This is the Bowman's capsule and these are the blood capillaries which lies in the cup cavity of the Bowman's capsule. These are called as glomerulus. There are two arterioles which arises from the glomerulus. One is the efferent arteriole and second is the efferent arteriole. The arteriole is the terminal branch of the renal artery which enters into the Bowman's capsule and it is called as efferent arteriole. The arteriole which leaves the cup cavity is called as efferent arteriole. The diameter of this efferent arteriole is always greater than that of the efferent arteriole. The main function of this glomerulus is ultrafiltration. Second part is the Bowman's capsule. Bowman's capsule is the cup-like cavity which is double walled and both the wall contains of squamous epithelium. The outer wall is called as parietal and the inner wall is called as visceral. The space between these two walls is called as the capsular space. The visceral wall contains specialized cells which are called as porocytes, which are small fetal-like structures which are important for the filtration, and they have filtration slits, that is small spaces present in between them. They continue into the neck and then the neck continues into the PCT, that is proximal convoluted tubule. The neck has ciliated epithelial cells. The third part is proximal convoluted tubule, that is PCT. PCT is highly coiled part which is lined by cuboidal microvilli, or it is also called as brush borders. Function of PCT is selective reabsorption. The PCT being the coiled part, passing of waste is through slow movement and that is the reason why maximum reabsorption takes place in the PCT. Fourth part is loop of Henle. Loop of Henle is the U-shaped structure which has two limbs. One is the descending limb and other is the ascending limb. Descending limb is thin walled, permeable to water, while ascending limb is thick walled and which is impermeable to water. The surrounding of vasa recta, the surrounding of loop of Henle with the help of blood capillaries is present, and these blood capillaries which surrounds the loop of Henle are called as vasa recta. That is, the surrounding capillaries of the loop of Henle are called as vasa recta. Function of loop of Henle is first to operate counter current mechanism which is important for osmoregulation of the body, that is for important of salt and water balance in the body. The next part, that is the fifth part, is DCT. DCT is distal convoluted tubule. It is also the highly coiled part of the nephron. It is simple cuboidal epithelial. Function is tubular secretion, that is waste secrete into the passing urine. Reabsorption and regulation of pH of body fluids. Last part is the collecting tubules, which is the long straight part of the DCT. The work of collecting tubule is reabsorption of water and secrete protons. The collecting tubules open into the collecting ducts. There are total two types of nephron. One is the cortical nephron, other is the juxtamedullary nephron. The cortical nephron has usually short loop of Henle and most of the nephrons are of this type, that is cortical nephrons. Second is juxtamedullary nephrons which has long loop of Henle. The 1/4 of cardiac output is usually supplied to the kidney. Now we are going to see urine formation. There are three steps which are involved in urine formation. One is ultrafiltration, second is selective reabsorption, and third one is tubular secretion. We will see step by step. First, ultrafiltration, which takes place in the glomerulus. Blood flow with high pressure in the glomerulus, and that is called as GHP. That is glomerular hydrostatic pressure, which is 55 mmHg. Next, it is opposed by the osmotic pressure of blood, which is 30 mmHg, and it is also opposed by the capsular pressure, which is 15 mmHg. So total the pressure which opposes the glomerulus is 30 plus 15, that is 45 mmHg. Therefore, the net filtration pressure or the efficient filtration pressure is 10 mmHg. 600 ml of blood pass through each kidney per minute. Therefore, glomerular filtration rate, that is GFR, is 125 ml per minute. Second one is the selective reabsorption, which takes place in the PCT, that is the proximal convoluted tubule. Here both ways, active and passive reabsorption takes place. Active reabsorption is from lower to higher concentration with the help of ATP, investment of ATP, and passive reabsorption is from higher to lower concentration without any requirement of energy. Active reabsorption takes place of glucose, amino acid, vitamin C, Ca++, K+, Na+, and Cl- ions. And passive reabsorption takes place of water, sulfate, nitrate, etc. 99% of the glomerular filtrate is reabsorbed by the selective reabsorption in proximal convoluted tubules. Third one is tubular secretion, which is also called as augmentation, which takes place in the distal convoluted tubule. Here actively absorbed waste like K+, H+, creatinine, and they are absorbed and they are secreted in the lumen of DCT, that is distal convoluted tubule, and in the lumen of CT, that is collecting duct or collecting tubules. The main work is to maintain the homeostasis, that is the constant internal conditions of the body. Example, in marine bonification and in desert amphibians, this is the only mode of Example, in marine bonification and in desert amphibians, this is the only mode of excretion. The next point is concentration of urine. We are going to see the concentrations of urine in different regions and the osmolarity. In cortex, the osmolarity is 300 per liter. In outer medulla, it is 600 per liter. In inner medulla, outer region, it is 900 per liter, and in inner medulla, inner region, it is 1200 per liter. As we know that in loop of Henle there are two limbs, one is the descending limb, other is the ascending limb. In loop of Henle, flow of tubular fluid is in the opposite direction, that is the counter current mechanism is operated here. And the vasa recta, which is the blood capillaries which surrounds the loop of Henle, also has two limbs, ascending and descending limbs, but their work or their directions of flowing of fluid is in the opposite directions. Ascending limb reabsorbs Na+ and Cl-, while descending limb, the tissue fluid concentration becomes increased, and that is the reason why water moves out of the limb, that is tissue fluid concentrates, and therefore to maintain the osmotic balance, water moves out of the limb of the descending limb. Now we are going to see what happens in the vasa recta. Before that, this osmotic gradient is essential for the maximum reabsorptions which takes place in this region, and that osmolarity or that reabsorption or that osmotic gradient is maintained by the vasa recta, because the osmotic gradient in vasa recta is exactly opposite to that of the Henle's loop. Osmolarity is 300 per liter, which is opposite to the loop of Henle's limb, means the here ascending limb is adjacent to the descending limb of vasa recta, and descending limb of loop of Henle is adjacent to the ascending limb of vasa recta. The next one is composition of urine. Color of urine is pale yellow color. The pigment is urochrome. Hypothalamus of the brain has osmoreceptors which can detect the change in the osmolarity of the blood. For example, if the osmolarity increases, ADH, that is antidiuretic hormone, is secreted by the neurohypophysis, that is the posterior pituitary, which stimulates reabsorption of waste from DCT, distal convoluted tubule, and from CT, that is the collecting tubules. Decreases the osmolarity. Due to ADH, that is antidiuretic hormone, the osmolarity is decreased. Here osmoreceptors also decrease once the osmolarity is decreased, and the secretion of ADH stops, that is antidiuretic hormone is now not secreted due to the decreased osmolarity, and this is called as the negative feedback mechanism. Next one is in the absence of antidiuretic hormone. If antidiuretic hormone is absent, it will lead to diuresis, that is dilute urine will pass, that is more water will be eliminated out of the body, and the condition will lead to diabetes insipidus. The another mechanism is RAS system, that is renin angiotensin. Another mechanism is RAS system, that is renin angiotensin aldosterone system by the JGA cells, that is juxtaglomerular apparatus cells. Blood supply decreases, then JGA cells secretes renin. What does this renin does? It converts angiotensinogen, which is secreted by the hepatocytes in the liver, and converts it into angiotensin 1. Angiotensin 1 further gets converted into angiotensin 2, which is the active form of that. Here angiotensin 2, which is the active form, has three main functions. First of all, it contracts the blood vessels, that is why it increases the blood pressure. Second, it enhances reabsorption from the PCT. Third, it stimulates adrenal cortex to secrete aldosterone, which is also responsible for the increased reabsorption of the water. Now we are going to see role of other organs in the excretion. First is skin. Skin performs diffusion of ammonia. They have two glands, one is the sweat gland, other is sebaceous gland. Sweat glands are usually situated on all the parts of the body, they are present everywhere, but they are most commonly or abundant in the palm and facial region. The function is thermoregulation. Sweat is produced to maintain the temperature of our body. Excess water, it also excretes water, NaCl, lactic acid and urea. Second is the sebaceous gland. Sebaceous glands are present at the neck of the hair follicles. They secrete the oily substance which are called as sebum, which protect the skin from injuries, or it also protect the skin from infection. Third one is the liver, which we have already third one is the lungs, which we have already seen that the spicy substances which we eat, or the spices which we eat, it has volatile substances, and that substance is excreted with the help of lungs. The next point is disorders of the kidney. First is kidney stone, or it is also called as renal calculi. There are different types of stones produced in the kidney. First is calcium stone, which are made up of calcium oxalate or calcium phosphate. Second are the strovoid stones, which are the response to bacterial infection, and due to the response to bacterial infection, these types of stones are produced. Third are the uric acid stones, which the people who don't drink enough of water and consume high protein diet, the stones which are formed are called as uric acid stones. Fourth type is the cystine stones, which are the genetic disorders. Now, what is the diagnosis of stones? First, you can check the uric acid content of the blood. You can also check the color of urine. You can also perform X-ray of kidney, sonography of abdomen, anything. Next is uremia. Normal blood contains 0.1 to 0. Next is uremia. Normal blood contains 0.01 to 0.03% of urea, but this concentration, if it is above 0.05%, then the condition is called as uremia, which can lead to kidney failure also. Third one is nephritis, which is the inflammation of kidney. This is due to the change in the osmotic pressure, and it leads to the movement of fluid from blood to the interstitial space. The next disorder is renal failure, that is kidney failure. There are two types of renal failure. One is the acute renal failure, and second is the chronic kidney disease. In acute renal failure, there is decrease in the urine output, but in the chronic kidney disease, there is decline or decrease in the glomerular filtration rate. That is the reason why it reduces the kidney size also, and it can lead to severe anemia also. Next is hemodialysis. What is Next is hemodialysis. The renal function, if it falls below 5 to 7%, then there is beginning of accumulation of harmful substances in the kidney. That is why there is the need for artificial technique or artificial mean of filtration, which is carried out by hemodialysis. Blood is taken from the renal artery, it is passed through the cellophane tube, which is coiled tube, and it is passed into the dialysate. Dialysate contains the same composition as that of the blood, but it does not have the nitrogenous waste, or it does not have the waste substances. That is why waste move from higher concentration to the lower concentration in the dialysate. Now blood is filtered and blood is returned back to the body by the vein. Lastly, here anticoagulant is added, which is heparin. That is the reason why the blood which is outside the body does not coagulate, or it does not stops flowing. Lastly, kidney transplant. Here the kidney is transplant to the patient from the suitable donor. It may be related or non-related donors, but the blood group matching and every tests are carried out to transplant the organ, that is kidney, from the suitable donor to the patient.